I should, yeah, and we're meeting now.
Yeah.
Okay, so yeah, just send it to the folder to all of us and then we'll find it, because your times are funky also.
Yeah.
Oh, and that link too. Do you want it?
Would you please send me the email, because I will.
It might not be, well, I can try to do it right now.
Yeah, and you can also, just remind me to say, hey, check the links.
Oh, okay.
It doesn't have to be specific.
Okay.
How are you, Dr. Dow?
I am spectacular. How are you?
I'm doing very well, thanks. Thank you for being with us last night at the Cowan Dinner. That was fun.
That was great fun.
Yeah. Hey, Donna, how are you?
Good, how are you?
All right. I do, yes.
And
then I put them up on YouTube?
Like, that's still
They'll just do it.
Okay.
Adults don't talk.
See you.
Yeah. I know, but I feel like you...
Do you want to get together tonight or tomorrow?
Yes.
How about tomorrow? Yeah, let's tomorrow at 6 tomorrow. Yeah. Same as always, we just go to the 11. Maybe not till midnight this time. No, I'm just late. Yeah, I do. Yep.
We have one new handout on the front table. If you'd like to grab one, please.
I got a new phone last night and I was like, I hope this is magic. I'm like, it's so random version of my I would know what to do. Well, I don't the time she gets home. Because usually like the 11th
We have one handout on the front table if you'd like to grab it.
Thank you. I mean, I'm pretty sure Dr. K would do the same for me because she knows that I'm neuro and that I need it. Right. Are you taking it? Nope. No, you already did it, right? No, I'm taking it this time. Right. Well, I'm going to take it next semester. Okay, yeah. Perfect. Yeah. Are you doing neuro? Third to last year? Yes. Okay. You gotta do it. Yeah.
I had 20 for today.
Yeah. Hey, Gabby.
So we have 17 today and 11 in the other one.
11 for the other one? Yeah. Yeah, that was me at the beginning of the psycho part today. I was like, I don't even know.
Yeah, no. I think we should be fine. We'll just like rush the people. Just rushing the room. Go.
Yes, we have one handout in the front of the room, please. Gabby is here. Yeah, that's the guy. You're fine. You're good. Yeah. Hi, Liz.
Hi.
Liz, we have one handout this morning.
Oh, thank you.
This afternoon.
There's how was your theater class? Yeah. It was not very hard. Like not hard, like the project. Like a little bit. It wasn't like you didn't like have to like. Yeah, it was because of dance class called somatics. Yeah, but even when I was like, I could do that. Yeah, stretching's good. I'm thinking we should do because we have yeah, I tried Well, I reminded Gabby like a second ago. So Gabby, I was just going to
Between the feeling again, or 17 again, that's going to equal to
Yeah. Okay. Gotta remember. You'll have to remember. I don't want to be dealing with like green. Yeah, I know. That's why I was like, I gotta remind you because I don't want to look weird. I honestly would have forgotten. I'm glad you reminded me. That's always what you do.
Hi, Alex. Alex, we have one handout.
Okay, so we're doing biotech and where else? I only need 45 hours, so I'm going to take some. I might just do that. It's on Mondays from 5:30 to 9:30, so it's for doing that. It is, but it's only the second half of the semester. Like it's 2 weeks.
Hello, Jess. Yes, we have one handout if you're just coming in.
5:30 on a Monday. That's my only like. Yeah. Okay. But
Excellent, Jess.
Long, but like it's not going to be hard. Pretty fun activity.
Alexa, we have one handout.
I just need to look into the for just some random two credit class. A lot of them have someone I know in PSYC. He was like, fuck what, he's going to do it too. And she's like, I'm going to take one thing instead. I was like, really? Like, I need to take you should take it with me. Yeah, I think so too. Yeah. So, when they both sent it, I never had drugs. I used to Kennedy. I need to ask them. I like living style.
Okay, we've got 12:30, so why don't we get started? Good afternoon, S&P.
Good afternoon.
All right, that was an enthusiastic response. Welcome to the last week of Vision. We'll be doing vision-related phenomena all this week, and then next week we'll start a brand new sensory modality. We'll be talking about audition, or the sense of hearing. So I look forward to wrapping up Vision with a bang. We have several ideas to talk about looking at our syllabus. It's always good for us to look a little bit ahead in the schedule. And so here we are on October 26th. And what I wanted to do was have a writing handout for you today that's actually way ahead of time. So it's actually more than a week in advance, but sometimes students like to have even more than 1 week. So I thought we'd just go through that. We'll be on hearing, and then we'll work our way back through our schedule to today and setting ourselves up for the exam on Wednesday. So I wonder if we can get somebody to read for us, just so we're all on the same page here. Sarah, I have this for you. I wonder if we can have somebody read through the instructions. Okay, thanks, Liz.
Do you want me to start with the citations?
Okay, so we can do the citations, and then subsequently, that's okay. So let's go after the citations.
Okay.
Subsequently.
Subsequently, write an essay that addresses each of the following. 1, using approximately 250 words from your best argument supporting Sharon and Candy's decision to seek a death for a moment. 2, using approximately 250 words from your best argument for why Sharon and Candy should have instead sought a hearing for a moment. Using approximately 250 words, form your best argument supporting the Galata students' demand for the resignation of their hearing president, Dr. Elizabeth Zinder. 4, using approximately 250 words, form your best argument against the
Gallaudet, yeah.
students' demand for the resignation of their hearing president, Dr. Elizabeth Zinder.
Okay, can we do the paragraph in yellow, and we'll stop after that.
Okay.
Address these points.
these points in the order described above, but do not number your responses. Instead, your essay should smoothly transition across the above stated issues. Your grade will be based on the quality of your writing style, i.e. grammar, clarity, succinctness, organization, and transition, and on the novelty and persuasiveness of your approach.
Okay, and then everything from thereafter is just formatting related issues, but I wanted to make sure that we got that out there. Some really interesting challenges that are rearing their heads within the context of our power and justice requirement. So we'll be talking early in that week about some of the physics of sound, as we did last time around. Remember that we had the physics of light and the facts of light. We'll do something corresponding to that for sound, and then we'll talk about the anatomy and the physiology. But this exercise gives us some idea of what it is to be differently situated socially because of one's endowments with respect to hearing in this case. I think it'll be really interesting. We've done something like this in more recent semesters that I've taught this course. On the flip side, we have a page that you're very familiar with. This is the feedback on writing style, and some students of mine who I have in multiple classes are extremely familiar with this. But I always provide this feedback to you in addition to ideas on your persuasiveness or your novelty. If that's particularly unusual, I'll comment on that, but I'm always looking at this. I think the greatest gift I can give to you in this class maybe is writing in a manner that's clear and succinct and in accordance with this. Okay? So I look forward to getting those. Again, this is going to be a ways out. Here we are on October 26th. This is not going to be due until the end of the following week, but at least you have it now and you can begin if you're so inclined. I think you'll find the readings to be absolutely fascinating. Okay, if we can work our way back in time just a little bit, over here I'll be doing the anatomy and physiology of sound and hearing and the properties of sound from a physics perspective. On Friday of this week, we'll be having another TED Ed on an article published by myself and Michael Votter, who graduated not that long ago. Does anybody know Michael Votter? Okay, several of you overlapped with Michael, a relatively recent graduate. So he and I, in the summer of 2011, worked on this report, and then over the next few months we wrote it up and we submitted it and it was published. And it does relate to visual attention, which we'll be talking about today. Okay, so before we get into today's conversation, just one more time, when we come in on Wednesday, right, we're going to be having our third exam, and we have out on the S drive this information, as we always have. This will be a mostly short answer kind of exam, driven largely by the TED Ed questions, which are based on the PowerPoint lecture notes, which are based on the readings. Okay, and we've done all of this before, so nothing particularly new, but I just want to remind you about all of that. Okay. On Wednesday, I'm going to do something a little bit unusual here. Just as a reminder, for those of you who are in our research class, that's the majority of this, but not everybody is in the research class. Just a reminder, because I might not see you later on today, that when you come in on Wednesday for that class at 1:30, the draft of your intro is due. And this is available to you on Blackboard. We talked about that last Wednesday, but it's been several days, so I thought I'd just give you that reminder. Is the research group okay with that? Okay, no surprises there. That's right on track with the syllabus. Okay? Yes, please.
What references in our intro?
You don't have to. You should be supporting your introductions with references, but I don't really care whether you have the reference list on that day or not. Okay? All right. All right. Really good. Okay. So let's do this. Let's get back into where we were on our last session. And we had a really fun session. We had a bunch of demos on Friday. We were finishing one of our last features in a visual sense, and that was the feature of depth. And we were talking about such issues as the poverty of the stimulus, and the fundamental problem in depth perception is retrieving the third dimension. So we actually had done this, but I just want to give you a little bit of a reminder because we did this at the very end of the session, and sometimes that doesn't get drilled into your neural networks in the same way that other information does. So the motion parallax phenomena is a very interesting one, and one that we ended our conversation with last week. Can we all get into position? I'll put my hands behind my back. The position that we use for our motion parallax display. Okay, people are lining that all up. Okay, and let's take a look one more time using only our right eye. So this is a monocular function. Okay, and why don't we have somebody explain to us as we're getting into position what they're doing with their thumbs? Anybody want to? Why don't you explain to me? Can somebody explain to me how I should get into position for this demo? You're basically doing that. You tell me. Go ahead, Clara. Thank you.
You're going to extend your right hand.
Right hand? Okay.
And line with the M.
Okay. All right. Very good.
And then go ahead and put your left hand about halfway.
All right. About halfway. Okay. Got it. Now what happens here?
Plant your thumb
Okay.
and the M.
Okay.
Close one eye.
Uh-huh.
And then move your head side to
Okay, move my head side to side. And I'm fixating on which of my thumbs?
The middle one.
I'm fixating on the middle one. And what I'm noticing is that the near thumb, as I'm moving my head, the near thumb appears to be moving opposite to my head motion and relatively quickly. By contrast, the distant M is moving with me and at a much slower rate. How many people are having that experience? Is that working for us? Okay. So that was the last demo that we did on Friday, just a little reminder about that. Okay. And then an idea that we had is that there are people who have maybe only one working eye, or maybe they have two working eyes, and yet they're unable to see stereoscopic depth. And I have brought it here in case anybody wants to take a peek toward the end of today's session. I do have a stereo tester. So you've probably done this maybe in an optometrist's office. You put on these kinds of glasses. They give you a small book that has things that pop up at you, and you have to say which of the stimuli is up the highest. Has anybody done that before at an optometrist? Okay. Well, if you want to check whether you have stereoscopic depth, because not everybody knows that they do, we can check you out later. But this will be a monocular cue that we have. Motion parallax. And the idea is this, that you're essentially, as you're moving your head from this position to that position, you're almost doing the geek trick as we talked about for photography. Can somebody remind us what the geek trick was for photography? The geek trick. Okay, why don't we go with Madison?
You take a picture of what you're looking at, and then you take the same picture 6.5 centimeters distance.
Okay.
And then you can refuse them, and it will print in 4D.
Oh, in 3D, okay, right. And Sora also had her hand up. Was there anything you want to add to that, or was that, okay, very good. Why 6.5 centimeters? That was a peculiar number that Madison had checked. Okay, go ahead, Zach.
It's the distance between your eyes.
On average, right, that's the distance between the eyes, okay. We also saw that one of the Mars rovers actually had two projectors, or two cameras rather, when it was up on the surface, and they slapped a blue filter in front of one of them and a red filter in front of the other. And so this thing is now roving around Mars doing the geek trick, essentially, right, and having these two cameras and projecting that all the way back to here to planet Earth so that we can see the surface of Mars in 3D, okay. If you don't have two working eyes, you can begin to recover that information by moving your eyes to these two positions. Who's following that? Is that working for us? Okay. So we had done that on Friday. I just wanted to make sure that one got into your brains one more time, because sometimes we lose the very last item. Okay. So for today's main event, we're talking about visual attention. Okay. And let's go to this. Okay. This is, I hope, a very interesting paper for you. Hope you enjoyed it as much as I enjoyed this paper. And it's from MIT. Okay. And we have a couple of ways to start out. Why don't I go to TED Ed questions, so we can get a list of the TED Ed questions. We'll work our way on through these. Okay. Maybe I'll go back this way, and we'll put any one student on the spot here, and then go to the think section. And our very first question was to describe the difference between top-down and bottom-up phenomena. Somebody want to help us out with that? Meg's been trying to get in for a couple of questions, so go ahead, Meg.
Top-down phenomenon is more knowledge-driven.
Okay.
So you can use kind of context clues
Okay.
to figure out the signals. And then bottom-up phenomena is more salient, so it's stimulus-driven.
It's stimulus-driven, okay? Stimulus-driven, very good, very good. So that's a really nice encapsulation of that. This is knowledge-driven, and this is stimulus-driven. And I think we've done this display before, so I apologize for the redundancy, but we have something like this. And then we have, right, I'm pretty sure we've done this one before. Say it with me as I go from left to right. This is a, and this is a, and this is a, okay, somebody's got the shtick back there. Okay, and this is a, and a 14. Okay, right, so this is the same stimulus, the same physical stimulation, but in this context, the top-down context would tell you that it's a B, and over here, the top-down context would tell you that it's a numeral, the numeral 13. Okay, right? So maybe I didn't draw that quite so well, but that's supposed to be a B. So it's the same physical stimulus on your retina, and so the bottom-up information remains constant, but we put it in two different contexts, and we have a top-down effect, right? It's knowledge-driven. Okay? So that's the very first question, and that sets the stage nicely for connecting back to something that we learned probably in Intro to Psych, top-down versus bottom-up, and something that we've got now coming to us that's new from the MIT lab. Okay, let's see if we can do this. We'll practice a little bit of neural navigation. There are these commonly used neuroanatomy terms. There's ventral and dorsal, anterior and posterior. Okay, and which of these four neuroanatomical terms best describes the frontal lobe? Anybody want to yell that out? Okay, go ahead. Gabby. It's the most anterior, right? The most frontal of all of these, right? The closest to our nose. Okay, so I think that's a good description. Can we all yell out which one of these four neuroanatomical terms best describes the parietal lobe? Dorsal. All right. Okay, really good. Can you all show me your dorsal fin? We think we've done that before, right? Did we do neural navigation this way? Okay. All right. Can you show me? This will be interesting. Can you show me a mid-sagittal slice? I think we've done this one. Okay. There you go. Yep, I see it. People are doing the mid-sagittal slice. Okay. Right. Can we go slightly lateral on the sagittal slice instead of it being midline? Let's go over to one side. Right. So this is perfectly mid-sagittal, and I can have still a sagittal slice, but a little bit left or right of my center. Okay. Can we do a coronal slice? It's been a while since we've tried a coronal slice. Let's think about that. I'm seeing several people doing a slice that is actually an axial slice, not a coronal slice. Coronal is this way, so we're wearing our crown in the coronal slice, okay? And you can take a relatively anterior or posterior slice, okay? Here we can take, if we're doing it this way, we're going to get a sagittal slice, and I can go relatively lateral or left lateral or right lateral, okay? And then we can have an axial slice, also called a horizontal slice. One more, yep, okay. And we can take that relatively dorsally or relatively ventrally, okay? Remember, it's doing all of that. Does that all sound familiar? Okay, it's been a little while, but it helps us, I think, when we're talking about these folks at MIT who are trying to understand what visual attention is, how there might be a neural basis between, a neural distinction between what is top-down and what is bottom-up, and they begin describing all of these things. Okay, let's take a look at this. I think this is a fun way to make a connection to our earlier chapter on consciousness. So how does the biological information in the article's abstract connect with the chapter on consciousness that we had from Dr. Nick Lane? Anybody want to see a connection? It's been a long time since we've had that conversation. Chapter on consciousness. We'll go with Jake this time.
Well, they're focusing on like neural firings, and they do like the hard problem of consciousness.
Okay.
Which is how do you go from that firing to feeling?
Yeah. Okay, excellent. How do you go from firing? That's what they're doing at MIT, right? They're going from these electrodes that are implanted, they're measuring firing rates. How do we go from that to something like consciousness, or if you will, maybe a surrogate for consciousness, which is visual attention, okay? So they're making that connection. Somebody else want to add to that? Was there, maybe Zach's got another one. Another connection.
I thought of it as like the consciousness article differentiated between like primary or like core consciousness and extended consciousness.
Okay.
And I guess I kind of saw a parallel between the core consciousness and just the bottom up.
That's excellent. I actually hadn't thought of that, and I've done this one for a couple of years now, and nobody's made that connection before. But thank you for reminding all of us. It's always good to review. There was core consciousness and extended consciousness, and you saw a parallel between top-down and bottom-up. Can you tell us which one goes with which?
So like the core would be more related to bottom-up, since they're both just like focusing on natural, like physical stimulation.
Yeah, right.
The top-down is more related to extended. It just requires more, I guess, like thinking and kind of.
Yeah, right. You have to somehow get out of your immediate environment through extended consciousness, right? So, and it's easy for me to ask you to think back to something that you learned in high school, but for a lot of animals, we couldn't do anything like that. We couldn't ask them to go back in time and recall explicitly. Presumably their extended consciousness is not that good, but I can try to call your attention to a particular event that happened a while back. Okay, and so I think that's a really nice tie. Something else. Well, maybe there are other connections that people had between the consciousness article and. Okay, Claire's got one.
I think along the same or similar lines that, that the bottom up and top down were sort of parallel to consciousness and unconsciousness.
Okay. Okay.
In the way that they're just two separate. Or the bottom up seems to be like a reaction, and it's done unconsciously. And
okay.
The top down is sort of like you're thinking about it.
You're thinking it through, right? You're deliberating. Okay, very good, very good. Something else. Lots of connections here. If you remember, there was a little ditty in that article about neural handshaking. Does that sound familiar? Neural handshaking? Yeah? Any neural handshaking going on in this this article? We've got the MIT article that we're reading. Yeah, go ahead.
We talked about like the neural synchrony and how it was like the neural synchrony between the frontal and parietal lobes were like stronger in low frequencies during top-down processing and stronger in high frequencies during bottom-up processing.
Okay, right. Who remembers doing the demo where I had differently colored magic markers and I would hold them maybe vertically or horizontally, and some of you were playing neuron, and you were horizontally tuned neurons, and you would go something like this, or you would clap or hit your desk or something to indicate how fast your firing rate was. And then I'd change the orientation, and a different population of neurons would come on board. And you would do the same thing for color. Right? We had some people who were blue-tuned, some who were red-tuned. Does that all sound familiar? Right? And at some point, we were getting synchrony between, for example, the red and the verticals. So that would be the neural indication. They're handshaking, if you will, in time. They're synchronized in time, and this is how the brain might encode red verticals. Even though we might not have a given neuron that is tuned to red verticals, you have a red population, you have a vertical population, and when they coordinate, we have that handshaking, we have that synchrony, and this might be how attention is coming together. Okay? Who's following that? That all sound okay? All right, so I think that's enough connection. Thank you all for your creativity in finding some of those connections. Okay, just for fun, we won't spend a lot of time on this, but I bet there are some really cool facts about the rhesus monkey. I've got a fun fact too. Maybe Liz has one. Go ahead.
I read on the National Geographic website that because of rhesus monkeys, doctors were able to discover the different blood types.
Oh, okay. I didn't realize that. I was aware there was monkey work there. I didn't know it was rhesus. Okay. Very cool. Some other fun facts that we have on rhesus monkeys? Meg's got one?
They're regular swimmers.
Really? I didn't know that. Okay. Okay. Oh, okay. A lot of primates are not. And I've been told that some chimpanzees actually establish their hierarchy, who's the boss, by who's least afraid to go into the water and get wet, because they're generally afraid of that. And if you can do that, you're not afraid of anything. But that's chimpanzee, that's not monkey. So, okay? Something else. Some other fun facts that we have. Zach has one.
Because it's the first animal launched into space.
Okay. Yep. Yeah. Okay, here's one that I can tell you. It was probably in the late 1990s. A used macaque was going for about $25,000. If you want to know what a used car is, right, or a used motorcycle, well, a used macaque monkey was going for about $25,000 back then. So what do we mean by a used macaque? There are people who do the kind of work that our friends at MIT are doing. They're trying to understand visual attention or some other aspect of neuroscience. This happens to be visual neuroscience, but they're doing some component of neuroscience, and they're recording in the monkeys, and then sometimes they need to get a brand new monkey that's naive, at least naive with respect to a particular task. So you might get one from another lab who'd been trained to do other tasks, but wasn't trained to do maybe a visual search, or wasn't trained to do a memory task, or whatever the particular task might have been. And back in the late 1990s, they were going for about $25,000. So you can imagine they're fairly pricey. And this is very, very highly regulated by the government. If you do have a primate facility, there are all kinds of rules you have to follow to make sure that the animal is safely maintained. Okay, that was really a trivial fact. Okay, then we have the pop-out displays and so forth. Okay, we don't need to go to all of that. Why don't we take a look at some of these? Let's go to, well, we'll be in figure 1 first. Okay, so we'll do figure 1 for a while. Okay. And maybe I can make that a little bit bigger. Can I zoom in? I'm going to go to a 150. Okay, why don't we see if we can get most of that in. Okay, can somebody, this might be entirely obvious, but can somebody sort of walk us through the sequence? There are two different kinds of trials. One is called the pop-out trial, one is called the visual search trial. Can somebody help us out with the sequence? Alexa's got that.
Okay, so on the pop-out trial, it's in both of them, you're looking for the same target stimulus, which is the green line that's kind of
that one? And that one?
Yeah.
Yeah, okay.
Mm-hmm,
Okay.
And in the pop-out one, it's called that because the target stimulus pops out at you. It's pretty apparent, and that's because it's a different color and orientation from all of the other lines. And in the visual search one, it only differs in one of those 2 ways. It's only a different color or a different orientation.
Okay.
And so it's more difficult to find. You actually have to look at each individual one before you can determine.
Okay. Right. And the monkey is earning their living frequently. They only get rewarded for food while they're doing this. All day long they're fasting, and the only time they can get their food is to perform well on these kinds of tasks. So they're fixating here, and their responsibility is to make a saccade. Can somebody tell us what a saccade is? To yell it out? It's a rapid eye movement, okay? So we can move if I ask you to follow my hand, you can have a smooth eye movement as you're following my hand, or you could look at me and then you could dart your eyes over to that window and then back to me and so forth. And those are actually slightly different neural signals, and people who measure eye movements will be typically specializing either in smooth pursuit eye movements or in these ballistic saccadic eye movements that we have. So the monkey is going to move its eyes only after it gets a cue to one of the four quadrants, indicating that one's my response. Right? So they're making their response not by pressing a button, as sometimes monkeys will do. We can train them to do that. They just make their saccade. One other point that was very beautifully described by Alexa, one other point I would like to mention is that if we think about this, the stimulation starts here, and then we get the sample, and then the stimulation goes away. If the monkey had not been paying attention, or if the monkey had his eyes closed during here, he could still do the pop-out task, right? Because there's enough bottom-up information that says that's the only one. The only possible answer here is that one. It's a singleton. So even if you weren't paying attention, okay, the environment alone provides enough stimulus-driven information to indicate which one is correct. Okay? So that's the bottom-up case, and that's why we say that visual pop-out is bottom-up. By the way, it sounds funny, pop-out, but that's actually what scientists call this thing. People will write NIH grants, NSF grants, and they use the phrase pop-out. Sounds very informal, but that's what, and it's because it pops out. Okay? So that's what goes on there. Down here, let's say the monkey zones out, has his or her eyes closed, and they come here. Now, if you're the monkey and you see this, it's not entirely obvious which one it is. Right? It could be any number of these, and there are other displays where it would be even more ambiguous. The only way really to pull it out of here is to have attended to that one and then go searching for it. Okay? All right. So that's really good. Let's take just one more idea down from here. Okay? Can somebody walk us through what's going on in these displays? We're still in figure 1. So what's this all about? What's a good take-home message from these guys? Okay, Meg's got something.
The history show that reaction times for search and pop-out differed significantly.
Okay.
So pop-out reaction times were faster than search reaction times. And there's a leftward shift for the visual pop-out in the standard signifying lower reaction times.
Okay.
And there's more rightward standard for the visual stick.
Really good. Yeah, very nice. It might not be obvious, but this is relatively leftward placed. This is relatively rightward placed. You can see 272 milliseconds versus 233. Kind of interesting. I mean, there's only a 40 millisecond difference there on average. But as these things go, that's enough to be statistically significant, especially if you have a few hundred trials. And one of your TED Ed questions was driven by something over on this side. I asked you to draw in Draw 2 a scatter plot. One of the items on the scatter plot had a slope that was 6 milliseconds per item for pop-out and then 22 milliseconds per item for the search side. Okay, so they're playing around with different kinds of distractor numbers. And we have a group in our research class that's doing that same kind of a thing. So they've got the steeper slope and the shallower slope. It's pretty interesting that across species we can see this kind of a pattern. Who's following all of that? Okay? Okay, really good. So we've zoomed in and we've looked very carefully at feature number 1, and we had you draw a whole bunch of items for feature number 1. For figure number 1. And that set the stage, I think, pretty well. Okay? Now, what I've found through the years, let me dial that down just a little bit, is that people really struggle with figure 2. So why don't we go ahead and take a look at figure 2?
Hey!
Hey, what? Okay, so that was my plant. Okay, those folks are in my earlier stats class, and I asked them at 8:30 this morning. When I say, "Why don't we take a look at figure 2?" I asked them all to yell, "Hey!" Okay, so hopefully you were up here, you were looking at me, and the environment came out and grabbed you. Okay, they grabbed you by yelling, "Hey!" So you are about to start fixating over here, okay, and use your top-down information to pull out something that is entirely knowledge-driven. And while you were attempting to do that, the environment grabbed you with a bottom-up stimulus. In this case, it happened to have been an auditory stimulus. So thank you to the 4 people who did that. I do this every year. I try to get somebody to, 1 year I blew it. I asked one student to do this, and another student overheard me, so I try to grab them in the hall or something like that before we come into class, okay? So they grabbed you with some kind of pop-out. It happened to have been an auditory stimulus, okay? We're going to see in a moment how they grabbed you by taking a look at the different brain patterns, okay? But here we are in figure 2, and you don't have to yell, hey, again. Okay, so here we are. And we're going to look at these two. By the way, you can forget figure 3, but there's a reasonable chance that information related to figure 1, figure 2 could easily come on Wednesday's quiz. Figure 4 could also be a good possibility. I will not ask you about figure 3 on Wednesday's exam. Okay? Somebody want to walk us through a main feature that they see down here in C and D? It's kind of subtle, but maybe you've got that. Why don't we go with Melanie again?
It's sort of like the times in the saccade is like reversed for pop out versus search. So for both of them, the LPFC is in the middle, but for the pop out, the blue sort of crosses the P is less than 0.05 level before, like further to the left, and in the search, the red one crosses first.
Excellent. Okay. So there's a lot there. That was exactly what I was looking for. Thank you, Melanie. I do want to make sure that we're all on track with this. There's quite a bit of information here. Maybe I can go even 1 or 2 notches bigger because we only need to see C and D. So let's see if I can make that a little bit bigger, and this might help, especially the folks in the back, okay? Melanie, that was spot on. I just want to make sure everybody is okay with all of these. So we mentioned we talked about the parietal lobe and the frontal lobe before. This is the lateral prefrontal cortex, is what we have there. Can we all point to our prefrontal cortex just to make sure we all know? Okay, so we're way up here. In neural anatomy, when you hear the prefix pre, okay, when you hear pre, that means more anterior. So not only are we in the frontal cortex, we're in the prefrontal cortex, okay, so we're way, way forward is where these electrodes are. And that activity is going to be measured here in green, okay? The lateral intraparietal is the other bit that they're measuring here, LIP. Can we point to our parietal lobes just to make sure we're on? Okay, so that's roughly where these electrodes are, okay? And then the frontal eye fields are also in the frontal lobe. They're more or less over the eyes, okay? So we have these two guys that are really quite frontal. The LIP is quite a bit more toward the back, or the anterior portion is also really quite dorsal, okay? So we're doing the pop-out task, okay? Or I'm leaning into this and say, why don't we take a look at figure 2, and 4 students yell, hey, right, okay? So now they're going to grab our attention, and what you might see happens first is that LIP, shown here in blue, sticks its head up first, and this is when we're first starting to get some synchronized firing that's greater than just chance level firing. So let's just make sure that we're all okay with that. Can we all go like, let's all start clapping randomly at any particular pace. Yeah, just go ahead. Just clap randomly. Okay. And now some of you, can you join me? Some of you? Okay, some of you are coming on board. And when they yelled, hey, that means that those neurons that were in the lateral intraparietal cortex came on board first. They began to synchronize at some level of synchronicity that was significantly greater than chance. In fact, it was 2 standard deviations greater than chance. How many people remember hearing about z-scores, maybe in some of your classes? Depending on who you had for intro to psych, you may or may not have had z-scores. If you had me, I'm all about the z-score. So you would have learned all about z-scores then. But now you're 2 standard deviations better than chance. So initially there's this glug, glug, glug that's unorganized. Then up comes the stimulus. And if it's a pop-out trial, we see the first synchrony emerging here. Okay? And then we get subsequent synchrony in the frontal lobe areas, which would include the lateral prefrontal cortex and the frontal eye fields. Those come on board in that particular sequence. So Melanie had that just right. Who's following that? Okay, and I'll draw a schematic of that for a moment, in a moment on the board. Let me go over here to the search task, okay? And now what we see is that this green and this red one, which are very frontally placed, are synchronizing. They're getting better than chance performance. They're now 2 standard deviations better than chance, relatively early-ish, and then after they've synchronized, the blue one comes online. Okay, then the parietal lobe finally catches up. Okay, so in one of these cases, in the case of pop-out, it's parietal first, and then we go to the frontal lobe. In this other case, for search, it's frontal first, and it goes to the parietal lobe. If we think about what we learned about the visual pathway, the first thing that happens is we catch a photon, the cis-trans isomerization. People remember that one? We're going to have that information go from our retinas back to our thalamus, all the way back to the visual cortex, back to the visual cortex. That's going to feed forward first to the parietal lobe, and then finally up here to the frontal lobes. Okay? Now, if you're looking for something, okay, your mom might say, go find the keys in the next room. Okay, now you've got knowledge about keys, but you're not in the next room yet. You're going to use some frontal lobe knowledge about keys to go looking for something. You're sending a signal from here back to the parietal lobe. Presumably that will begin to activate also the primary visual cortex. Okay? So a really interesting change in what's synchronizing first, depending on whether we're in this type of attention, which Zach had linked earlier to core consciousness, versus this kind of attention, which he linked earlier to extended consciousness. Who's okay with that? Is that working for us? I think that is exquisite. By the way, for many years, maybe even a couple of decades before these folks at MIT put these electrodes in, we knew behaviorally all about pop-out, and we knew behaviorally all about search. Those were not new. We've had that information before, but we didn't really know how the brain was making that distinction. We knew that we had steeper slopes, a really steep slope over here for search. We knew that this was very fast, right? But we didn't know what the underlying neuroscience of it was, and now these folks have unpacked this for us. Okay? Yeah. Alexa has a question.
I'm a little bit confused.
Okay.
So in the pop-out task, we said earlier that it's more bottom up.
The pop-out task is bottom up.
Is bottom up.
Yeah.
Okay. So because it's
it's entirely stimulus driven, right? Yes. And yeah. Does that work?
Yes.
Okay.
And then this is showing us that the parietal lobe is activated
first.
So
Uh-huh.
the parietal lobe is sort of what we're connecting with, like starting the bottom up?
Yes, right. Even though it's quite a bit later than here and here. Okay. After we get to the visual cortex, what happens? Well, it begins to feed forward all the way, and it can't get all the way there to the frontal lobe until it hits the parietal lobe first. Okay? Does that work?
Yeah.
And then, if mom says, go get the keys in the other room, and you're not yet in the other room, you have knowledge, just like you have knowledge about the alphabet, you have knowledge about what keys are like. So now this thing is cooking, and it's going to send stuff back from the very, very top of the visual pathway back to the more rudimentary. Okay? And it didn't have to be that way, but they grabbed it in MIT. I thought they just nailed this. This, by the way, is a science paper. It's very hard to get into science, but when you have these kind of data that are giving us the neural correlates of what we do all day, every day. Somebody in class yells, hey, okay, and your brains did this. Okay, well, they've got it here beautifully laid out in MIT's lab. Okay? Okay, let's go on to, let me just build on that. I promised myself that I would draw this as I do every year. We're going to try to draw just the schematic of a very crude brain, which I'm going to draw like that. That's your brain, and the brain is now facing that way. Okay, so your occipital lobe is back here. Right, occipital lobe, this will be the frontal cortex, or FC, and you might want to take this down. Okay, and this person is looking that way, and you're looking at it as if you're looking at my head, so there's a lateral bit here. So we can draw this kind of lopsided brain. Parietal lobe is going to be here-ish, right? And frontal lobe is going to be here-ish, right? And frontal cortex. Okay. So what happens is when the animal is doing a pop-out task, there is desynchronized activity at first, and then at some point, some of the neurons begin to respond, and they respond first here. Okay? And as we'll see in a moment, they respond at a relatively high frequency. So what I'm going to do is I'm going to go from here, I'll draw a line up, I'm going to draw something like a sine wave. It originates in the lateral interparietal cortex, and here's my arrow, and then it lands this way. It's going to go like this. Okay? It's going from a relatively earlier place in the visual pathway to a later place. Okay? From the bottom to the top, and this is going to be the pop-out case. Okay? Now we can use a very similar display and have the same rules make us a CAD to the target, but now we're going to have a visual search display where you actually will need to attend to the early stimulus, the sample, so that you can make the response. And I'll draw something out this way. It's going to originate here in the frontal cortex. It's going to be a much lower frequency, which I'm drawing like this. And it will start there and then come back to the parietal lobe. Okay, so this one is search. So it's really the same two critical areas of the brain, and we get neural synchrony going on, but the sequence is different. That's one thing. And they're also different with their characteristic synchronized frequency. One is relatively lower, and one is quite a bit higher. Okay? Which I think is really, really intriguing. Who's following that? Okay? And we're going to see if we can have somebody help us unpack that. I tried to call your attention first to this figure. This is figure 4. And I was going to have you especially focus on these PSYC items that are for pop-out and for search. Here's a PSYC pair. Let me turn the lights down to give us some better contrast. PSYC means the neural activity around the saccade, just before or just after. And they're plotting here the coherence level, and they're subtracting away the intertrial interval coherence. So we're getting a lot of coherence here and a lot of coherence here. How is this one different from this one? Somebody help us out with that? Okay, Claire's got something.
The coherence is switched. So the pop-out coherence on the left-hand side is much lower than the other, or on the right-hand side and vice versa.
Okay, so perfect. Okay, so we're going to go blue is low, red is high, okay, as we see over here, and then blue is high, red is low. Okay, I'll do that one more time. If we go from blue to red, we're stepping up over here. Is that okay? Blue to red, we're stepping up. And if we go to the corresponding bit over here, we'll forget about all this just for a moment. We're going to go from here to here. We're going to go down. This is up. This is down. Who's with me? Does that work for us? Okay. For those of us who have taken Psych 200, that would be a classic crossover interaction. And we're getting a classic crossover interaction. And what's interesting here is that this is the level of coherence that we've got. We're stepping up from blue to red when we're in a particular kind of frequency range, 22 to 34 hertz. Okay? We might call those, as they do here, the middle frequencies. Okay? Then we can go up to considerably higher frequencies, and we're getting blue and red. So we're getting more blue than red high-frequency stimulation, and that's more high-frequency stimulation for pop-out than for search. Who's following that? Okay? And so that corresponds to, sorry to take the lights up and down all this time. Okay? Here's the high-frequency bit, and here's a lower-frequency bit. Yeah? Okay. All right. Oh, Meg's got something.
We're going opposite directions?
Yeah, so blue to red, and blue down to red.
So that's
Oh, yes, and so on the search case, we're going to be starting for the visual search. For those of you who do this in Cog Lab, this would be your conjunctive search. Okay, that's what they're calling search. A little bit confusingly. It starts in the frontal cortex and goes back to the parietal cortex, and it does so at a low frequency. Or actually, they're calling it a middle frequency, but it's lower than this high frequency one. Okay, and that's why that's drawn the way that it is. You still look a little confused. We'll try that again. It's okay?
For the search, we started with frontal cortex and
We start at the top and we go down, because it's a top-down search.
then for the pop-out,
We start at the bottom and we go up. Yeah, right, okay, yep. And we go up at a relatively high frequency. It didn't have to be this way, but this is the way apparently our brains are encoding all this. Okay, let's do one more. Okay. Can somebody help us out with this bit? This is the last of the graphs that we've got. It's actually number 4 again, but it's a different aspect of number 4. And I'll try to make that as big as I can so people in the back can see that comfortably. Okay. Somebody want to give us an idea of how to interpret this? It looks moderately busy. Okay, Liz is right on it.
So with the lower frequencies, there's more coherence in the visual search.
Okay.
And as the frequencies get bigger, there's more coherence in the pop-out.
Excellent. Okay, so one key here is to recognize that this is frequency, right? This is lower frequency, this is higher frequency. If you could hear these frequencies, it would sound something like this. You'd be going from these low frequencies to these high frequencies if you could play them, right? Okay, but this is how many times these cells are firing per second in a synchronized manner. Okay, and we're getting at these lower frequencies, this middle band is happening, and we're going to take advantage of this. So thank you for that. More coherence in search. Okay, so this is again going to be top down. Okay? And then over here we have the complementary pattern. These dotted lines are showing how far you'd have to go for the synchronized firing to be significantly different from just random firing in the brain. Okay? So now we're in these middle or upper frequencies. They're relatively speaking upper frequencies. And as we can see over here, we're getting more coherence in pop-out. Right? Okay, so that's the pop-out, and that's going to be this higher frequency band that you see here. So hopefully you can connect the drawing that you just made to figure 4. That might help us clarify all that. Let me turn those in the opposite direction. All right. Who's okay with that? Okay, well, congratulations on making it through an electrophysiology paper from MIT. I think that's not a trivial task, okay, but you did it. And I think it's just beautiful how systematic that is. And we got a little preview into this even in the first week of class when we learned about neural firing and synchronous neural firing, different kinds of consciousness. Right? And here it is in all its glory as measured by electrodes in some animal that's not too distant a relative of ours. As other animals go, these rhesus monkeys are primates just like you and I are primates. Right? So we would have had a recent ancestor with them on the order of a few tens of millions of years ago. That seems like a long time, but you could go much further back than that for other kinds of animals. Okay? Questions or comments on any of that? Okay, Liz has something.
Can you briefly go over what a z-score is?
Okay, thank you, Liz. I would love to, because that's one of my favorite topics. I'll try to contain myself and see if somebody else wants to tell us what a z-score is. Sarah's got a z-score.
It's just a score that indicates the number of standard deviations a certain raw score is away from the mean.
Yeah, thank you. Very, very nicely explained. Okay, our stats students do those all the time. And maybe we can go to, where else do we see a z-score? Okay. Yep, so here we just have random firing, and we want to see if your firing can get synchronized at some level that's better than chance. As you're clapping randomly, it might have been the case that Claudio just happened to have clapped at the same time that Sibel clapped. So they would have been sync, but they were synchronized only by chance. Okay? Is there some moment where we can say, you know what, actually, neuron Sibel and neuron Claudio are firing at a rate that is so synchronized, it's probably better than chance. And if they get to a certain number of standard deviations better than chance, we say go, and a standard deviation is a z-score. Something else. It was a great question. About any of this. Okay, Alexis.
So when you talk about higher frequency versus middle frequency, that's just referring to like the frequency of the, what are those?
This is synchronized neural firing.
Okay, it's neural firing. Okay.
So can we do quarter notes? I'm gonna go with quarter notes. Can we have the people over on this side join me in quarter note clapping? And can you stay with me on that? I'm going to stay here. Can we have people on the window side? Can you do eighth notes if you would, please? Can you stay with me? Okay. So we have two different synchrony rates going on. We have something that is lowish. Okay, thank you, neurons. Wow, what a beautifully synchronized set of neurons. So that lower bit that we had over on this side would be the neural firing that would start with search.
Okay.
Okay?
Cool.
Right. Very good. Very good. Do that one more time. That was a lot of fun. Okay, and I wonder if we can do it this way. Why don't we pretend that this room is your brain, okay? And that's the back side of the brain, and this is the front side of the brain, okay? So what we're going to do is I'll be the metronome, and why don't we see if we can get a pattern. Well, we'll make this entirely a search pattern, and we'll have the people who are more anteriorly placed join me first, and then people toward the back of the room will join me later, okay? So here's the synchrony. Okay, then after a couple of claps, the next row back does it. And then finally, after a few more claps after that, the signal has finally gone from the frontal cortex all the way to the back, okay? Then 4 crazies in the room yell, hey, okay? And we get a faster signal. Can you double me with eighth notes? There are the eighth notes. But the eighth notes start from the posterior portion. Stay with me? Okay? Way back there, and then they make their way up front. Okay, so it's going to roll forward. There we go, okay? All right. Okay, so that's the synchronized firing at relatively slower rates, quarter notes, or relatively faster rates, eighth notes, going either front of the brain, back of the brain, between those two. Top to bottom, bottom to top. Yeah? Boy, they're elegant there. Something else. No? How about we do this? anything else from this section since our last, well, we have a couple of minutes, so anything you've really been dying to ask, we never quite got to this question or that question over the last several sessions, maybe about the Pulfric effect, maybe about random dot stereograms. We did some form perception before that. Go ahead, Maddie.
With the visual imagery, is that kind of similar to
wow, that's going to be...
about the keys and like the top?
Okay, right, yeah, so it is going to be the case that we do get area B1 lit up here, right, but it's not going to be immediate, right, because we're going to need the frontal cortex to say what it should be remembering, okay? So I think
Go to the occipital lobe instead of the parietal lobe?
it would probably, there might not be a direct flight from here, because you probably have to make it through there and then back this way, okay? But yeah, the final destination will be the occipital lobe, and you probably have to go through the parietal, right? No direct flight, probably. Great question. Something else? A layover in the parietal lobe. I love it. Something else. Go ahead. Yes, thank you, Madison.
When we were talking on Friday about free phasing
Yeah.
and random dot stereograms,
Okay.
how are, so when you have two images, I'm looking specifically at this side here.
Yeah.
If when there is no disparities,
You're flat as a pancake when there's no disparity.
so
Okay.
then, are they not identical when you have the selected area move?
That's exactly right. What we do is we just generate random patterns, okay? And this is going to be your right eye's view. Okay, it's backwards. This is your right eye's view. We're going to move it to the left in your right eye's view, and that's going to make that small hidden square pop out at you and come nearer to you because we're crossing in that case, right? And then we just backfill the area. We will have slid this one little box over and we'll backfill it with some kind of random black-white noise, right? Okay? Yeah. Very good. Something else? Okay. All right, we'll give you a minute off for good behavior. Thanks for a good session and for making it through an electrophys paper. Folks who are going to be in that 1:30 class, if you're running participants today, I'll see you. If not, if you want to turn in your 1:30 papers, you can for that class. Everybody else will see you on Wednesday for the exam. Okay? All right. So I think groups 1 and 2 are running today, right? Okay? Right? That's how we're doing 1, 2, then 3, and 4.